Hi everyone. Just gonna wait for a few minutes for folks to join. Yeah, just gonna wait a couple more minutes for them to join.
It's all good. Everything at MIT is 5 minutes after the hour.
Yeah. Yeah, it's sort of the standard time. But it's still trickling in. I think maybe we can start now. So I have the great honor to introduce Alex Shallett to give the Rose Weiner Levin Lecture today. And this is a special seminars in oncology lecture funded by the Rose Weiner Levin Fund. The Rose Weiner Levin Lecture was established by the late Rose Weiner Levin and her children, Michael Weiner and the late Marjorie Bloom, to further research within human cancer genetics. The Levin family remains committed to accelerating improvements in healthcare for patients and their families, and we are grateful to the Levin family for their long history of supporting Dana-Farber's dual mission in cancer research and care. So thank you for that. And this is a particularly special lecture because we have with us, I think, a really great speaker, Alex Shallett. And many of you won't remember this time, but there was a time, when telephones had rotary dials and cars didn't exist yet, and single cell sequencing was not a thing. And that's actually sort of when I met Alex back when he was a postdoc in Aviv Regev's group, in addition to co-mentoring by Hong Kong Park. And it was really he that at that time spearheaded the development of single cell RNA sequencing, which is now transformed cancer research, but then I would say all biomedical research. He was a postdoc from 2011 to 2014. Previous to that, he got his bachelor's his master's and PhD degrees at Harvard and bachelor's degree at Columbia. And he's now an associate professor at MIT with appointments at the Broad Institute as well. Alex continues to develop really creative and useful technologies to interrogate tissue heterogeneity. And it applies them to a variety of diseases, including cancer, but then also infectious disease and immune diseases, which, for us cancer folks is especially interesting nowadays when immuno-oncology is so important. So I'd like to introduce Alex and look forward to hearing a great lecture.
Thanks, Ramin. Thanks so much for the kind words. It's still weird to be introduced, but thank you all for having me and thanks for joining. Let me go ahead and share my slides. And so I'm sorry that I can't be there in person. It's always much more fun to talk to people and have dialogue afterwards instead of staring at my screen and having a conversation with it. But
sorry to interrupt. Sorry, I should say, I meant to say that when folks do have questions, please don't put them in chat. Please put them in the Q&A thing so that they can be compiled for Alex to answer at the end.
Great, cool. I appreciate it. Usually I like being interrupted. We can do it that way as well. So thanks so much for having me. It's a pleasure to be invited to give this seminar. And what I was hoping to do today was to tell you a little bit about some of the work that we've been doing collaboratively with a number of other labs around the Boston area, trying to identify and rationally modulate cellular drivers of cancer response. Sorry, somehow I got in there twice. And I wanted to tell you a story that I have yet to tell in public yet, so I'd love to hear feedback and thoughts on it. It's really focused on pancreatic cancer and understanding the role of the microenvironment in dictating tumor cell state. But before I begin, I sort of just wanted to frame broadly the kinds of problems that my lab is interested in, what we study in general. So broadly, my lab is interested in how balance is achieved in tissue ecosystems. And I know, given this audience, that I don't have to introduce how important balance is for homeostasis. If you have an overexuberant immune response, you can get tissue damage and death. And on the other hand, if you have too little of an immune response, you can become susceptible to opportunistic infections or the outgrowth of tumors. What my lab is really interested in studying is what is it that defines balance in these systems? What are the deviations that are induced by disease? And how can we think about therapeutically rebalancing them or buffering them in the first place? And to do this, we spend a lot of our time developing and applying tools to really understand these complex multicellular ecosystems. In the context of cancer, the way that we've been going about this is through a nice, supported project through the National Cancer Institute, U54, a cancer systems biology consortium, that's really trying to think about, the quantitative and functional mechanisms of resistance in cancer, and sort of divided into two major projects, one that looks at cell intrinsic mechanisms of resistance and one that looks at cell extrinsic. And I totally recognize that these are coupled and we'll get back to it, but, trying to separate those two out. In terms of what we're doing in the overall grant, we're bringing together a number of different single cell profiling technologies to study cancer that include innovative methods developed in the Manalis lab to think about single cell mass and cell growth. And let me get a laser pointer right here to make our lives easier. Perfect. Innovative platforms to measure cell mass growth rates, some strategies for doing single cell RNA sequencing, some functional assays in combination with some incredible models and primary samples that come out of the Weinstock and Hahn labs over at the Farber, as well as some computational work. And I'll be presenting work that really is a huge collaborative effort and spans a number of different PIs and labs. In particular at MIT, it's work with Doug Laufenberger, Scott Manalis, and Chris Love, and definitely could not happen without partners over at the Farber, including David Weinstock, Andrew Aguirre, Bill Hahn, and Brian Wolfman, who really have the deep domain expertise. And I'm showing you the 8 of us here, but really these are the people that are at the tip of the spear. Peter Winter, who's a fantastic postdoc between Scott and my labs, who will be on the job market at some point, and I encourage everybody to aggressively recruit him. Andrew Navia, graduate student, Mark Murakami, who now has his own independent lab at the Farber, and Tree, who's a fantastic postdoc at Bill Hahn's lab. And it's not really 4 people. I'm going to show you some work that really depended upon this incredible ecosystem of graduate students, clinical coordinators, doctors, research technicians. And so really, this is the kind of effort that takes a village. And something that I think is so cool about being in the Boston area is the opportunity to work in these large collaborative projects to really try and do translational science. So like I said, we have this large project that's really trying to do systems biology in the context of cancer. And the story that I wanted to talk about today is some efforts that we've done to understand cell extrinsic factors that govern therapeutic resistance in cancer. And as I said, this is the first time I've ever presented this, so I look forward to all of your feedback. So the cancer in which we've been looking at cell extrinsic mechanisms of drug resistance is pancreatic ductal adenocarcinoma, or PDAC, which I'll refer to it from here on out. As many of you probably know better than I do, given your clinical training, PDAC is an incredibly challenging tumor to detect. It's very involved to remove, and very often people don't detect it until you have a liver metastasis, and it has an incredibly poor prognosis. Incredibly aggressive disease where if you catch somebody early, the best thing to do is to perform surgery. And the majority of people come in and already have metastases, very commonly to the liver. Genetically, it has a very distinct mutational profile that many of you may be familiar with common mutations in RAS and in P53. But surprisingly, there's still no targeted therapies that take advantage of these known genetic mutations. And that's sort of well captured in this slide that I'm sharing right here that comes from Andy Aguirre's lab, where they've looked at a number of different potential mutations, and despite this relative uniformity, we really don't have good inhibitors, and all of our efforts to try and block RAS signaling really haven't led to the interventions that we might like. So what that means is the pancreatic cancer field is really focused on two major things. One of those is really thinking about how you might use something else like transcriptional phenotyping to do subclassification, since the genetics don't seem to be quite as informative, and then some modeling, originally cell lines, and now increasingly complex systems like organoids to try and do personalized medicine, so that you could really try and find targeted therapies that would be specific to the individual. And so, on the transcriptional side, there's actually been a tremendous amount of work done by a number of labs. And here are some of the canonical papers in the field that have looked at bulk RNA sequencing and have defined signatures that associate with different classes of pancreatic cancer. And there have been a number of different classification schemes that break down in different ways, each of which has provided sort of stratification that has helped to describe a more aggressive form of the cancer and a less aggressive form. The one that I'm going to focus on, that I'm going to think about most, is this one right here, basal versus classical. It comes out of this paper by Moffitt et al. In Nature Genetics 2015. And the reason we're going to focus on is that it's going to turn out to be a little bit more applicable in the framework that we look at. And it's one where, rather than just looking at bulk, they tried to do some in silico microdissection to remove stromal contaminants. And so I'm sure that many of you are well aware, one of the biggest issues in pancreatic cancer is that actually it has a very low percentage of cancer cells. And so a lot of what you're looking at when you're doing signature analyses are, normal tissue or, non-cancerous tissue. So, as you see this, you can see that it appears that there's a clean stratification between this basal phenotype right here and this classical phenotype right here, with the basal phenotype being associated with a poor prognosis in the classical form. Now, you might ask yourself, when you look at this is a little messy. We're doing all of this in bulk. To what degree are these gene expression programs, sort of set in stone? Are they discrete? Or, as you sort of go down and you zoom in and you look at the individual tumor level, is this actually a spectrum that goes from the basal state all the way over to the classical with some sort of intermediate in between? And equally importantly, if genetics doesn't seem to be deterministic in a lot of this, what is it that is actually dictating cellular state? If it's not mutations, is it something about the epigenetics, or is it something extrinsic to the cell about maybe the architecture or specific interactions with other cell types in the microenvironment? And these are fantastic questions to ask, but you might say to yourself, how do you actually go about parsing these inputs and figuring out what is most important for these cancer cell states, to really perform some sort of translational work? And so what we've been working on in the context of the U54, spearheaded by Shri and Pete and Andrew and Hannah Williams, is an experimental pipeline that would enable us to begin to address these questions, where what we're doing is we're starting off with pancreatic cancers that have metastasized to the liver, where we're getting needle biopsies. And then we're taking these incredibly small amounts of material and separating them out into two pieces. One where we're going to create models so that we can go study them a little bit more mechanistically and do follow-ups, and one where we're just characterizing the cells directly out of the biopsy to understand what does the tumor microenvironment look like. And so this enables us to create a fundamental reference to understand the biology of these metastatic tumors, and a place where we can start to look at our matched models to understand to what degree the models recapitulate the biopsy and to begin to use them to test potential hypotheses. Now, it's nice when I put it on a slide like this, but a fundamental problem in all of this, as you know, is that a needle biopsy is not a lot of material. So you might be asking yourself, how can we go about, both getting a comprehensive profile of a sample and at the same time building matched models with which to study them? And this is where, some of the stuff that we've done in the past in collaboration with others really becomes critical. And that's because over the past several years, there have been these transformative advances that let us take low input samples. And I don't know why my screen's freezing. So for example, to take a complex mixture, isolate individual cells using micro devices, whether they are multi-well plates, microfluidic circuits, reverse emulsion droplets, or microwells, then perform reverse transcription to capture all the messenger RNA that is being expressed in a cell. And from that, to look at these cells in this incredibly high dimensional space where we're looking at the expression of 20,000 plus genes. Here I'm showing you it's 3 dimensions because it's hard to represent 20,000. To figure out where cells sit in this high dimensional space, we usually reduce this by doing some simple dimensionality reductions like this t-SNE right here, where now each point is a cell, and I'm showing you groupings of cells based upon their similarity in this high dimensional space of 20,000 genes, and then coloring based upon some computational approaches that say that each of these cells are more like one another and dislike one another. What that lets us do, as you well know, is go and find genes that are specific to each of these populations using simple statistical tests. And from that, we can extract what are the components of these complex cellular mixtures and really identify, what are their attributes. So we might find a set of cells that expresses TCR, which tells us they're T cells, and we might further go on to ask questions about whether they are proliferating, whether they are exhausted, and so on and so forth. And really, what's enabled these complex systems to be dissected are these increasingly, sophisticated technologies that build, on advances in micro and nanotechnology. And the one that many of you are probably familiar with that has enabled a lot of this work are methods based on droplets. And really, there was an incredible set of papers that came out in 2005 that described the original formation of these methods. And so there's a method called DropSeq that we played a small role in with Evan McCosco, Steve McCarroll, Oni Basu, Aviv Regev, Dave Waits, Hong-Kyun Park, Rahul Satija, and others, and Josh Sains. A parallel method from Mark Kirshner and Alon Klein called InDrop. And now there's a commercial variant, which many of you probably have used or routinely used, that sort of hybridizes the two, called 10x. And really, there were some foundational papers at this point that describe the idea of using these droplets as a way of processing complex cellular mixtures in high throughput. And each of them fundamentally takes a mixture of cells, isolates those cells with a barcoded bead, and then lyses those cells so you can capture the information, tag it, reverse transcribe, and build a sequencing library and figure out what RNAs belong to whom based upon barcodes that come in through these beads. Now, the question that you might be asking yourself is, great, can you go and apply this to your pancreatic samples? And that was the question that we had for ourselves. We have these beautiful, complex multicellular tumors where we want to understand what are the different components and what are they expressing, instead of just looking at 3 or 4 of our favorite markers. Can we apply some of these droplet-based approaches? And the problem was that they really weren't amenable to the kind of work that we wanted to do. And the reason for that is that they weren't really scalable. So it's going to be hard to run a tumor in an adjacent normal or to run a tumor in blood. They weren't going to easily move from our lab over the Farber. They weren't efficient in the way in which they process things. They load at low densities so that you don't get doublets of cells or doublets of these barcodes. But what that means is you very often get orphan cells and you're wasting material, which when you're talking about a needle biopsy doesn't make sense. You just don't have the material to get rid of. Finally, these things are expensive, and if you want to run a lot of them so that you get statistical power, you want to figure out how you can make it cheaper. And in looking at all this, we realized that there was no real technology that was going to solve all of the issues that we had. But fortunately, we realized that maybe there was a simple solution in the lab of one of our collaborators, Chris Love. And so Chris has pioneered these incredibly cool cellular assays based on this microwell array that I'm showing you right here next to a quarter. And so this microwell array is basically, think of it as a big ice cube tray. It's got about 100,000 small little boxes that are about 50 microns by 50 microns by 50 microns. And he's done a lot of really nice imaging and characterization assays in them in isolation of CTCs, and out there just like refer you to his work. But what we became curious about in chatting with him was whether we could take everything that was great about DropSeq, sort of this idea of pairing cells with barcoding reagents and isolating them, and moving it from something that was complicated into a really simple system that would work just on gravity. And this is work that was spearheaded by Todd Guerin in Chris's lab, along with Mark and Travis in mine. And I'll say that there are other people that have used microwell-based approaches in the past, and they all sort of work similarly in some ways, but ours is a little different, and I think helps with some of the clinical pieces. So this technology that we called seqWell for at least 3 bad puns works like this. The idea is we start off with a very simple array of small little boxes that we functionalize. We can now come in with the same beads that you would normally use in one of these droplet-based methods, but because our wells are roughly the same size as our beads, we can fill almost every single well with a bead, over 98%. What that means is if we now come and use a ball pipette to deposit on a cell using gravity, there are many wells that we will end up failing to load because we're going to load it, maybe at 10% occupancy. But every time a cell falls in a well, it will have a bead and we can capture its information. And then finally, and most importantly, we figured out how to seal these arrays with a semi-permeable membrane. You can think about this as a screen window. And basically what it lets you do is it lets you take this entire array, dunk it in lysis buffer, liberate all of the RNA, and then switch to a good high salt hybridization buffer, capture more RNA, peel it off, and do everything you would normally do. And this also helps deal with cross-contamination. So, I'll save you the trials and tribulations, and I'll just tell you that we spent a lot of time making it work, where we could get to a point where we got a similar number of transcripts and genes in mouse and human cell lines. But what really is transformative about it is that this is the entire rig. We now have a simple clamp. We have our array of wells right here, and we have a membrane that pinches it together. And you've got many in a box, and these are cheap and disposable. A fancy pipette, you don't have to use one. And here I'm showing you, as I said, a clamp from Agilent, but this is something that you could 3D print, or you could even use like a t-shirt press to seal many of these in parallel, which we have in the past. Now, nobody really cares about whether or not you can run a cell line in some of these things. And the question becomes, could you work with something that's, a little bit more complicated clinical specimen? And so what we were able to show in work I don't have time to describe here was that actually, in fact, this simple system would let you work with just about anything. And that's because you have a really simple array. You're going to just deposit a few cells by gravity on the surface. So you could work with complex mixtures like blood to characterize, responses here in hyperacute HIV infection. We could take small pinch biopsies that were half the size of a poppy seed, from regions of inflamed and uninflamed colon and ulcerative colitis, to look at cellular changes associated with that. And we could even take fluids where we didn't know if there were cells like cerebral spinal fluid from leptomeningeal disease patients and characterize the cellular content and responses to therapeutic intervention. And most recently, we've even pushed this a little bit further to take those same nasal pharyngeal swabs that you probably hate getting for your COVID test, and to use them to characterize both changes associated with severe infection and also the most likely targets of SARS-CoV-2 RNA. So what this means is that we have this system that will work with an incredibly small input. And that's fantastic, because now what we can do is we can go back to our original problem, PDAC, and start to think about actually setting up a pipeline where we take a core needle biopsy, dissociate it to get viable cells, and then go and create a sequencing library. And so what we did is we took these core needle biopsies, dissociated them, ran a targeted DNA sequencing panel to get CNVs and point mutations, and ran single-cell RNA sequencing on 23 different biopsies. From the cells that we get out, we run what's now a pretty standard analysis, a copy number variation analysis, looking at the average expression over windows of DNA in our single cells to infer changes in the amount of DNA or copy number variations. And we can use this to confirm which cells are malignant. We see good agreement with what we would get by the targeted DNA sequence. And this helps us to classify what are the malignant cells in each of our needle biopsies, and to also find the non-malignant ones. So we did this for 23 different metastatic PDAC samples. Here I'm showing you some colors that go with them. A few of them had previously been treated. The majority of them are liver mets, though we do have 3 other sites, for example, one from an adrenal gland and one from the peritoneum. You can see that the majority of them harbor the KRAS mutation that I've talked about in the past, as well as TP53 mutations. And on average, we're getting somewhere in the 1,000 or so cells out of each individual one. But critically, because we're loading so few samples onto this array, or cells on this array, we have enough material that we can actually go initiate organoid cultures, grow out those organoids, and perform single-cell RNA sequencing at early passages to figure out to what degree these are representative, and then to take them out further so that we can start to do functional assays with them. And so, this has enabled us to actually, get a pipeline that both gives us, good single-cell data, but also good formation of models with which to go do subsequent functional testing. And so I want to walk through a little bit what we're seeing in the data and some of the surprising things that came out of it. So first off, we can look at what's happening on the malignant side, and we can ask ourselves questions about how this aligns with what we've seen in the past in, these signatures like the basal and classical ones that I introduced before. When we go and do, our standard t-SNE, we can see, like in most of these things, that our clusters of cells separate by patient. Not surprising, given that each one sort of has its own developmental origins in its individual. Surprisingly, when we look at the markers for each of these, we find that one of them, actually appears to be a neuroendocrine tumor. And we went back and, shared notes with the clinicians, and they went and validated that this was in fact a neuroendocrine tumor. So it's cool that we can get this out of our sequencing data. It was just so different than all the others. It defined one of our first principal components. And subsequently, I'm going to remove it for most of our analyses. And so imagine that we really have, 22 that we're looking at. So you might ask, what is driving variation in our data set? And fortunately, actually, something simple like a principal component analysis captures some of the major axes of variation in our data set. So if we look, what we see is that our first principal component here strongly correlates with an EMT signal. Our second principal component right here is strongly associated with classical and anti-correlate with basal. So really PC2 defines classical versus basal using that Moffitt paradigm. And finally, our PC3 really seems to associate with cell cycle. And you can see that basal seems to be linked to EMT, and what we find is that there's a high and low EMT set of basal cells that I'll come back to. So we see this basal to classical divide. A first question you might ask yourself is, are all the tumor cells basal, or are they all classical? When we went and looked at the individual cells, what we found was actually something incredibly surprising. We see a number of cells that appear to be primarily expressing genes associated with the basal program, many that are expressing genes primarily associated with the classical program. But there's a distinct set of cells that are very clearly co-expressing, both sets of genes. And, we wondered if this was doublets, and we spent a lot of time looking at a number of different things, thinking about, the number of genes, the amount of mitochondrial reads we were getting, so on and so forth. But everything that we were seeing convinced us that this actually looked like a co-expressing phenotype. And so you might say, well, that's strange. Are there unique attributes that associate this co-expressing phenotype? And it turns out there are a set of genes that are very strongly associated with what I'm going to call these intermediate cells for right now. And so I'm showing you up here a series of the different genes that come in, like CX3CL1, VCAM1, that are strongly associated with this intermediate phenotype, where I'm plotting now an axis of basal to classical on this axis, and then how much of this intermediate score shows up. But what you see is that it looks like there's actually this tripartite graph that should have been worked by others as well, like Mario Suva and Ifai Tarosh, when they're looking at gliomas, where we go from one stream to the other, but it looks like maybe there's an intermediate transitional state. If we go and look at what's enriched in these genes right here, we see that this intermediate state seems to be strongly enriched for some developmental gene programs, RAS signaling, and KRAS, inflammation and stress. And we can ask, what do these cells look like more broadly if we go and look in the literature? And in looking at signatures, it really looks like they are similar to some normal pancreatic progenitor populations. So whereas basal genes and classical genes seem to line up with productal progenitors, really these IT genes seem to be significantly associated with this undifferentiated phenotype that came up in this atlas of normal pancreatic development. You might say, well, you're putting all this in basal and classical. Why didn't you use one of the other sets of signatures? And it turns out when we go do a deeper dive into these previous RNA taxonomies, that basal and classical actually capture a lot of the differentiation that we see. So if we look, our classical set of signatures actually seems to strongly correlate with a number of the other ways in which people have characterized these tumors in the past. We see that we've got our basal phenotypes down here that seem to correlate very strongly with some of these other signatures like squamous, et cetera, with significant overlap. And so if we look through all the various studies, we see that these line up very cleanly. But previous studies really hadn't seen this intermediate transitional, and this might be something that we uniquely got because you're now at a single cell level. So the idea that you would not have A or B, but you'd have a half A and B, you can actually resolve. And so, really what we get is we get this graph right here that tells us that maybe what we have is sort of a classical to a basal with some sort of intermediate, transitional state that really looks like an undifferentiated precursor. So now you might ask yourself, well, you have this tripartite graph. Where do individual tumors fall? Is it that, some of them are basal, some of them are classical, some of them are intermediate, or do you see a spectrum of states at the individual tumor level? Well, if we go look at the bulk averages of our data, what we find is that, across the 22, removing that one that was neuroendocrine, we see some that look like they're very classical here in blue. We see some that, look like there's sort of this intermediate phenotype right here. I have the neuroendocrine that's over on the side. And then we have two that are sort of basal that are really separated by how much EMT program they have. But when we go and look at what's happening within them at the individual cells, it turns out that every single tumor has a blend of classical, intermediate, transitional, and basal cells. So we actually find that many of them are dominated by one or the other, but we see every single, and we see all three states in each of the tumors that we look at. You might ask, is this something that exists in situ? Can you go validate it? Is it something that comes in? Remember, we're in the metastatic site. You might want to know, is this a primary pancreatic cancer thing as well? So, what we did, and this is really some incredible work by Hannah Williams and Shri and Pete and Andrew, where they basically went, with Brian Wolpin and Andy, and they basically went and took 10 cases where we had single cell RNA-seq. And they did multiplexed immunofluorescence for them as well. We picked, some marker genes for this, and then we tried to pick some markers for immunofluorescence to try and think about the relative fraction of phenotypes in each of our samples. And so basically we get a series of different immunofluorescence markers that are going to help us to define the classical, the co-expressor, or the basal phenotype that, are some of the major things here. So some keratins and, GATA6, which is a transcription factor canonically associated with the classical phenotype. We go image, and lo and behold, we see cells that look basal in a tumor, we see cells that look classical in a tumor, and we see cells that actually appear to be both basal and classical. And if we go and we do calling, for tumors, what we see by immunofluorescence and what we see by single cell RNA sequencing, we get cells that based upon, RNA appear to be classical down here, and ones that appear to be basal, and ones that appear to be co-expressors, and we see roughly the same proportions and the same ratios, when we look at it by immunofluorescence. And I think reassuringly, if we go and we look across each of our individual tumors in the 10 that we look at, 1 intermediate, 2 basal, and 7 classical, we see that there's a good agreement between the multiplexed immunofluorescence and the single cell RNA-seq, where it's really significant for the associations within a tumor, but insignificant for the cross correlations across them. And as we dive in a little bit more, what we find is that actually, as we go from tumors that are more basal to more classical, that there is an increasing amount of this intermediate transitional type, which really, leads you to this idea that maybe this intermediate transitional represents sort of a bona fide transitional state. So maybe what we have now is a spectrum that's plastic where these cells could move. And so you might ask yourself, with all of this heterogeneity that you see, what is it that drives such heterogeneity? How plastic are these? How much could you push from a basal to a classical, or a classical to a basal? And are there features, intrinsic, maybe some of these mutations that will really lock you into a specific state in your tumor? And so one of the ones that we wanted to start with to really get at these questions was to look at KRAS amplifications. And so KRAS mutations are ubiquitous, but amplifications have been, suggested in a lot of the literature to strongly associate with the basal state. And so a series of papers are out there that suggest that, really KRAS amplifications drive this particular phenotype. And so if we go look at our individual tumors and we infer, copy number variations, we can see that we have more KRAS inferred copy number alterations and gains in our basal tumors than we do elsewhere, and that's a significant increase. And we see, an increase in, these transcription factors, GATA6 and HNF4G, that have been associated with the classical phenotype over here. And when we go and we look at, where our individual cells fall based upon amplification, what we see is that, yeah, it looks pretty good. The KRAS amplified cells are primarily located on, this basal side over here, not classical. You have a couple that are over, but, it's primarily basal, and maybe a little bit intermediate transitional. And so you might say to yourself, well, is this a genetic event that locks you into a specific transcriptional state? Does it override everything else? And so this is where the organoids come in and play an incredibly critical role, because what we can do is we can keep the genetics the same and then go and look to see whether or not there's any phenotypic transition. And what's so surprising when we go and take these malignant biopsies and we take our organoids and we sequence and we look at them is that there appears to be an incredible shift in culture from really basal all the way over to much more classical. And this is for each of the individual tumors that we look at, and it's incredibly significant. So it really looks like there's an incredible amount of plasticity within these cells. And you might say, well, how are you sure that you haven't had some sort of outgrowth of a clone that's done something and loss of a particular clone? And that's because we can actually track some of the individual clones using our CNV variation. So if we really go in and look at exactly the same copy number variants where we have our KRAS amplification, what we see is that really we started off with about 200 cells in this biopsy that came early that were strongly basal, and later we see that the same mutation profile and the same copy number alterations are associated with a classical gene expression program, which suggests really that there is plasticity within these copy number variant clones. So the genotype is not locking you into a particular state. We can look more broadly than at our organoids to try and think about what is gained and what is lost, since obviously we're no longer actually phenocopying. And what we see when we dive into this is that for our particular tumors, we have a very good take rate of organoids in early passage, so sort of around P2. But as we go out further, we see that actually very few of our basal organoids establish. Our ITs do okay, but really our classical ones are the ones where we can really get these to propagate out for a long time. At a genotypic level, they look very similar to what they were originally, but the phenotype is strongly diverging from what it was in the past. And so when we look at, sort of expression of the genes that are associated with our basal cell state or our IT state or our classical state, really we have a strong expression of genes in our organoids that are associated with classical, maybe a little bit of IT and a major loss of these basal programs. You might ask, what are gained and lost when we go to these environments? So what we can do is we can try and now go look for genes that are differentially expressed between the biopsy and the organoid. So we do this for each individual pair. We take ones that are significant for a number of pairs, and we get about 103 genes. And what this leads us to is a series of different programs that are specific to our organoids and the way in which we culture them. They are genes associated with epithelial identity, oxidative stress response, and amino acid metabolism and the like. And so there really is like an organoid phenotype, and I'm just going to refer to this as organoid specific going forward. And so, basically, we've seen that these things are evolving, and you might ask, well, what's happening at the individual clone level? What's happening within your dish? Well, we actually went in and we did some longitudinal characterization of some of these organoids over time to understand drift and understand clonal selection. And what we find is that there does seem to be a drift towards this basal phenotype in the majority of our organoids over time. Here, I'm showing you each time that we sampled it. And for each of these, I'm showing you the fraction that assigned to basal, intermediate, transitional, classical, or that really just look like organoids. Some of these, fail to culture past a specific point. Some of them continue out. And for individual ones, we can look at what are our copy number variants and how they diverge and what are the phenotypes that are associated with them. So for example, for this IT phenotype right here, what we see is that we have one dominant CNV clone that starts out right here that's primarily basal and maybe a little bit, intermediate transitional. As we go on to passage 2, we lose the majority of it. It becomes more organoid specific. Down here, it comes back a little bit and goes forward and goes forth. We have what appears to be, some clones that grow out that are more organoid specific, or, that are more, or that come on that were originally organoid specific and become more classical over time, and that come on to dominate. And really, at the end of the day, even for things that look like they were more basal to start, we end up with more classical on the back end. In other instances, we do actually see that sometimes the genetics dominate. So I don't want to tell you that the DNA is not important, the genetic profiles aren't critical, because here, for example, we have one biopsy where we have a P63 amplification. We start off being primarily basal. Some of our clones decay out, and stay basal, and other ones, come on and really continue to become increasingly basal. And so there are places where genetic events can supersede the environment. But in the majority of instances, what we're seeing is that the microenvironment plays an instructive role in defining transcription phenotype. And so, you might ask what's driving this. And so we can now go back and for each individual class, so basal, IT, or organoid, we can see what's different between the organoid and between control. And what we see is that actually, not surprisingly, it's a lot of the environmental signals that you would normally have in vivo that we don't mimic in culture. So strong enrichment for things like interferon signaling, ECM, cytokine signaling, focal adhesions. And so we see that, really, there are genes that appear to be strongly associated with, say, basal, like enrichment of interferon signals that are coming in there, as well as with the IT, some adhesion that's associated with classical phenotype. And that really these are things that strongly distinguish our biopsies from our organoids in each of these individual models. So things that are specific to basal, things that are specific to IT, things that are specific to classical, and things that are sort of common among all of them. And so, you might say, well, these are signals that appear to be coming from the microenvironment. Can you figure out from the non-malignant cells what might be driving this? And so the answer is yes, we can go back and look at our data. And here is a t-SNE of our non-malignant cells. Here you get good interdigitation of cells across all of our different patients because the immune cells are common among them. We don't know much about the metastatic tumor microenvironment. What are the cell types and phenotypes? We might want to know something about the stroma and fibroblasts because primary PDAC is strongly enriched for them. And you might say, is there specific associations with the TMA? Well, yeah, we go and we find a bunch of cell types and states. There are about 18 of them on our data, multiple different fibroblast populations, inclusive of myofibroblasts, dermal fibroblasts, periocyte-like fibroblasts, and even one tumor that has some inflammatory caps, a couple different myeloid populations. We find that, interestingly, there's a very strong association between specific fibroblast populations and the metastatic site that we then go validate by doing some comparisons, some bulk RNA sequencing. But what I really want to focus on for this is sort of thinking about the environment and the signals that come from it. So let's go back to thinking about what may associate with these basal, classical, or IT phenotypes. So if we go and take these non-malignant cells and we sort of compare them across groups, what we find is among the basal tumors, there's this incredible enrichment for C1QC positive TAMs, that look very much like the ones that were described previously by Zhang et al. In colorectal cancer, and a real decrease in CD8s and CD4 T cells. So maybe it's a little more cold than you might expect, but not cold and not cold overall, because there is a huge immune infiltrate. It just happens to not be T cells. Interestingly, you can see here that these tumors are really dominated by this tumor-associated macrophage. And overall, what we see is that actually basal tumors are much less diverse along like an ecological index, something like Simpson's diversity, than classical tumors that appear to be more diverse and have multiple different subtypes. The intermediate transitional is actually strongly associated with T cell frequency. So if we look at the abundance of CD4s and CD8s, they're strongly increased in IT tumors. And just to summarize a lot of data that I don't have time to go into today, what we find is basal is this C1QC TAM high, intermediate transitional is T cell high, and that we have SPP1 positive TAMs that are high in our classical tumors, just as a rough idea of what's going on. So what we know now is that there are specific transcriptional phenotypes. They're associated with specific environmental signals relative to what we see in culture, and they're associated with specific cell types. So to summarize where we're sort of at right now, what we know is that our microenvironment influences our phenotype independent of genotype. We have, expression programs that are enriched in vivo that associate with like EMT, cell-cell interaction, cytokine signaling that are missing in our organoids, and that, there seems to be associations between our malignant cells and our immune cells, but, we don't know exactly which yet. And so if we think about, this, normally in our metastatic microenvironment, we have autocrine and paracrine signals that are important inputs to phenotype. We go into our organoids, we come in with various factors that drive, the generation of these outgrowing, and then we have some various signals and factors that we're adding to basically continue to propagate these out. And so if, our organoids are strongly getting this organoid phenotype and drifting, a simple question might be, well, what happens if you remove those organoid factors? Are they driving the tumors more basal, or is it more complicated than that? And so, interestingly, when we take out these sort of organoid growth factors like Wnt3a, correspondin, and these TGF-beta inhibitors, what we see is this huge shift where we go from organoids, so these tumors that were primarily classical to being increasingly basal. So a significant shift back towards the basal side. We also see a significant downregulation of those genes that I was calling organoid specific. And so, the cells are reduced a little bit in terms of their cycling, but they have stable CNV profiles. And you might ask, well, what does this look like at the individual cell level? Do you actually see a shift? And yeah, we do. So if this is where we were, at a particular point in our culture, we started off, primarily being classical, and now we're going to strip out these organoid factors and we're going to see what happens. And we see a transition to a place where we actually have cells that occupy all three states, controlling for CNV. And so what this tells us is that we can actually recover all three states ex vivo. Interestingly, when we go and study cell lines that people have used, for example, at the Cancer Cell Line Factory over the Broad, we see that the majority of them are basal. If we take those cell lines and we put them in organoid media, they lose their basal expression and become classical. The majority of organoids are classical. If we place them in cell line media, they become basal. So there's this incredible phenotypic plasticity that's possible by controlling the environment. And so the natural question is, if we can do all of this in a dish, are the factors in the tumor itself, in the microenvironment, playing an important role in defining transcriptional state? So we can go back and we can ask, what are the autocrine signals and paracrine signals that might be important, the things coming from the tumor cells themselves that are reinforcing a phenotype, and what's coming from some of these surrounding cells, those T cells or those macrophages that might be locking a tumor into a particular state. So we can go and we can look at potential soluble factors that may come from the tumor cells themselves that would dictate this spectrum. So I'm showing you a pretty complicated plot here, but I'll break it down really quickly. Here I'm looking at things that are more highly expressed in basal, things that are more highly expressed in classical. Here are things that are higher in our intermediate transitional. Here are things that are higher in our basal and classical phenotypes. And here I'm looking at things that are specifically associated with the Go term for secreted factor. And so we can rank a variety of factors by how much we think they impact intermediate transitional, classical, basal, so on and so forth. And so, for example, here we see TGF beta 2 that seems to be strongly associated with IT and basal relative to classical, which leads to this natural hypothesis that if you were to take one of these organoids and dump in TGF beta, you should be able to push this thing from being primarily classical to being over on the intermediate transitional / basal side. And so that's exactly what we see. You take this model, we start off being primarily classical, and as we add TGF beta, we shift through an intermediate transition over to more of a basal side. And you might ask, why do we care about all of this plasticity? The answer is that these cell states are incredibly important for thinking about how effective various therapeutic interventions are. So if we take what's the common drug that's used for PDAC, so gemcitabine, what we can see is that our classical tumors are incredibly sensitive to gemcitabine. If we now add in TGF beta to shift it over to this intermediate transitional / basal phenotype, we become orders of magnitude less sensitive to drug. And now if we actually just withdraw TGF beta and do exactly the same assay, we come back to right where we are. So what we're seeing is that actually the states that we put ourselves in phenotypically can dictate how responsive we are to standard of care. And so what I think this suggests is this idea that you could start thinking about modulating the environment to control plasticity within your tumors to try and alter the efficacy of drugs that you use. What about on the paracrine side? Well, remember I told you that T cells were strongly associated with the intermediate transitional phenotype, right? And so here you can see that interferon gamma is coming in loud and clear as being neither associated with basal or classical, but really high in this IT phenotype. And so we can map where this goes, and really the gamma seems to be coming out of the T cells in the microenvironment. So you might say, well, if I add gamma, is this going to push my tumor from classical, or my organoid from classical up to intermediate transitional or not? And once again, it turns out it works beautifully. You go and you take your organoid, you dump in interferon gamma, and all of a sudden you move from being primarily classical to being, this intermediate transitional type. And that's sort of where the center of mass stays as you continue to culture out. So you get this significant shift. And this is super interesting because remember, I told you that the basal phenotype is much more aggressive and worse than the classical phenotype. This is the one that has the better prognosis. And so you might ask yourself, if I was going to prescribe a checkpoint inhibitor that was going to reactivate the immune system, would I actually be doing something that wasn't good for this individual by forcing the cells into an intermediate transitional type and then down to basal? And so I can tell you, at least in an N-of-1 experiment, that's what seems to be happening. So if you take, a particular case that I show you here, where there was a local diagnosis and resection, a progression of a metastasis that was biopsied, you can see that it started off with the tumor cells being primarily classical. And then on immunotherapy, for a little while, a resection taken later, and you can see that there's a transition from classical to IT and basal. It really classifies primarily as IT when you look at the single cell level. And if you look at what's up in the relapse versus what you had in the primary, you see a strong enrichment of interferon responsive genes, a couple basal ones, and a real decrease in some of these classical gene programs, suggesting that actually this is a place where immunotherapy might lead you into greater progression. And this is all N of 1, but I wanted to put it out there, just to sort of help you think about potentially the relevance of some of this in vivo. So, what I hope you took away from this talk about PDAC is that distinct microenvironmental signals are critical regulators of PDAC transcriptional state and plasticity. That it means that it's incredibly important for us to think about the tumor microenvironment when trying to create models here, because it's going to influence the efficacy of the drugs that we select and the relevance of them for in vivo. And it also tells us something about the idea that we could actually try and modulate cell extrinsic factors to govern tumor cell phenotype to change therapeutic response. And so really where we're going now is we're trying to build on this. It's going to be a lot of different screens to try and figure out what are the boundary conditions for a particular phenotype, how does genetics overlay on top of this, what are the combinations of factors that we could use to block transition from classical through intermediate transitional back to basal, and really trying to control that to make our drug interventions a little bit more effective. I'll just give a quick plug to what we did in Project One, which I spoke about a little while ago, but I think it's super cool. I'll just say that in some work with David Weissock and Mark Murakami, we've really been looking at cell intrinsic features. Here, looking primarily about drivers of therapeutic response in BALL or acute lymphoblastic leukemia. In this general paradigm where you treat, you drive to minimal residual disease, and unfortunately, the majority of people relapse. What we find there to do an injustice to the story by making it too short is that, the ALL falls along the spectrum of normal development, but we see dysregulation of signaling programs. We can use this to identify gene signatures specific for each state and do better jobs of classifying. This becomes important when we go look at relapse, because it turns out that at relapse, you see that more aggressive tumors are more developmentally mature and are enriched for specific mutants. So you see more RAS mutants showing up in some of these tumors that are more developmentally mature, and they're the ones that come on sooner, where you have worse prognoses. And finally, what I think is really cool about this is because of this incredible patient-derived xenograft repository that David and Mark have set up, we can look at minimal residual disease, and we can actually characterize what are the pathways that are activated in these cells at MRD to prevent, to enable them to escape therapeutic pressure. And so we find programs that we find that, our MRD cells fall on this spectrum where some of them appear to be more differentiated in cycling and to be using BCR signaling. And we see a set that are less cycling and that are more quiescent and higher for stress response. And what that does is suggest combination therapies that we could use with, ponatinib, one of the, leading BCR-ABL compounds, to drive deeper remission than sort of two drugs targeting, BCR-ABL and a common mutation in one of its sites. And so, really quickly, I think that this shows, once again, this interesting coupling between genotype and phenotype, and helps us to think about how we can modulate phenotype, in therapeutic interventions to make our cells less viable. And I'll just say that more broadly to where Ramin started all of this, what my lab has really been interested in is experiments like this, where we really start off with human biology, working with partners all around the world on, infectious diseases, cancers, and autoimmunities. And then we try and take that data to think about what are appropriate models, be they organoids or, cell lines or PDXs, to find interventions that we could accelerate back into humans. A big part of this is not just running these experiments, it's also creating these large repositories of information, where we keep data that can be used to cross-analyze new data sets that come in and find patterns and things that already exist. And then what we like to think of as Rosetta Stones, which are, really signatures that help us go between data sets. And so to aid in this, and is something for the community, we've stood up a resource that for a lot of really bad reasons we call Alexandria, that basically pulls together all of our single cell libraries into a large library so that we can start to do analyses to generate hypotheses. So for example, during COVID, of what are the likely targets of SARS-CoV-2, but also so that when we bring in new data sets, we can understand how it relates to things that we've seen in the past to help us prioritize what models we may want to use. Behind all of this is a lot of information that we have generated looking at multiple different models in vivo and in vitro, information on different compounds and what they might impact, cytokines and chemokines, and a series of different signature data sets that we hope will just drive discovery. So I want to do 2 final minutes here and just sort of talk about where this all leads us. I think that, when I talk about some of the biology that comes out of this, I think it's super cool. And I think there's so many things that, we're going to be able to do with, partners at the Farber and the Broad and all around the community pushing forward on this stuff. But the other thing that it brings up to me are sort of the limitations in some of the experiments we've done. That there are things that I wish we'd been able to do better, so we could have gotten more out of these, samples that were very generously donated by patients. So, for example, one of the things we want is better data. We want to be able to measure more markers on our cells to figure out what the cells are secreting, since I told you microenvironmental factors are so important. And so, for example, one thing we've been working a lot on is how to get more information per cell. So, we recently released a manuscript in Immunity that I refer you to, where we basically created a new molecular biology to boost our gene and transcript capture to get almost to where best-in-class plate-based protocols are, and to do a better job of capturing things like transcription factors and cytokines. And I'll tell you that this works well for SQL. It also works well for DropSeq and for methods like SlideSeq, in case you're curious. One other thing that we've done that I think is incredibly useful for some of the clinical work that we're doing is we've developed a new way to basically simplify our clinical pipeline. So if we think about what we're doing, here I showed you some FNAs from the liver, so we might take a fine needle aspirate, then we need to cellularize to take those cells, load them on our arrays, seal our arrays, lyse our cells, capture our mRNA, pull them out, reverse transcribe to get to something that's stable. And in work that was spearheaded by Alex and others, what we wondered is if we could take these fine needle aspirates, pause, so load our arrays, freeze them, and then even ship them, and then come back and process later to do this centrally, because that would simplify clinical collection in a lot of different sites. And so we set up an experiment where we're actually now looking at liver FNAs from hepatitis as opposed to PDAC. Alex flew over to Rotterdam and ran some samples on site, where he ran all the way through sequencing generation, some where he loaded and then froze. And what he saw when he did this for both the fine needle aspirate from the liver and for PBMCs was that he captured similar proportions of cells in both, including for hard to capture cells like granulocytes. But most importantly, when you dive in and look at this data, what we see is that each individual cell population is represented in each sample, and that we have basically no differentially expressed genes. So we're getting equivalent data, whether we run all the way through or if we collect on site and process and bring back. I'll finish by saying that, where the lab is really thinking about going now, and what we're really excited about, is thinking about the role of compounds in some of these models, thinking about interventions. And we're working on new ways of doing increasingly complex screens in increasingly complex models like organoids, so that we can look at what a lot of different genetic or chemical perturbations do. And so happy to talk more about that. And the other thing that we're thinking a lot about is how to really understand the role that tissue structure and interaction plays in phenotype. I showed you how important some of that is in PDAC. And so we're building new platforms that enable us to think about these features and screen them. So let me finish by thanking everybody in the lab, who are all fantastic. I really want to, in particular, thank Peter Winter, Andrew Navia, and it's always confusing to find everybody on these slides. I want to thank Nola and Jennifer, who I will find in a second, as well as Michelle, who have all played a huge role in getting this stuff processed and getting this done, and all the work that I showed today. There's Michelle and there's Jennifer. And I want to thank all of our collaborators. This was a huge project that involved partners at MIT, at Brown, at Dana-Farber, and that wouldn't have been possible without that network of colleagues. So I want to thank our funders, who've been incredibly generous and hopefully will continue to be. And with that, I'll just say thanks and take any questions, and hopefully I'm roughly on time.
Yeah, I think it's almost perfect. Claudia, I think you have the questions, is that right?
Yep, you can just click on the Q&A tab at the bottom to read the questions from the audience.
Yeah, somehow it's not coming up for me.
It should be at the bottom of the screen.
Yeah, I'm pressing on the Q&A tab, nothing's coming up. Oh, maybe here it is. Okay, so yeah, so the first question was from Paul Doan, which is how can you make sure that one well? I think this is about your SQL technology, that one well has only one cell.
So it's a great question. So the way in which we do it is we load it in incredibly low density. So we make sure that every single well has a bead, but we load it at a density where we have, sort of on average 0 cells per well. So 100,000 wells, you might load 10,000. And so the probability that you get more than 1 cell per well is low. We then go on to do the standard things that everybody does in sequencing, where we would, sequence our libraries, look at some of the various quality metrics, look at some of the signatures to confirm that it is a singlet as opposed to a doublet. But I'll also say that one thing we can do, and we should do more of, but we don't always do because it's complicated, is we can actually look at the arrays and we can image to figure out what is the frequency of doublets in our wells. It's just, it takes a long time to scan over those multiple boxes. So if you're worried that you've overloaded, it's something that we can look at. And unlike another run on another technology, if you're unhappy, you can literally flip the thing over, dump everything out, and go at it again. It's one of the nice things about sort of a simple system like this that is visualizable, that works on gravity, and where the chip is relatively inexpensive.
And how many cells do you get per chip typically?
So standardly, we will get somewhere in the few thousand out. You can load more using some of the cell hashing strategies that you're probably familiar with. We do things a little differently. Our consumable costs when we make them per chip, we're sort of in the tens of dollars as opposed to the thousands of dollars. So we would rather load at a lower density, get a higher proportion of singles, and run several in parallel. And we've gotten pretty good at this. And for truth in advertising, Chris and I have started a company that is making these where it's even simpler to load than we ever could have imagined. And hopefully those will be coming soon. And I give a plug for that because I know that some partners of the farm are actually doing some beta testing, and excited to see some of that come through.
Great. So the a second question comes from Sydney Moyer, which is addresses the plasticity of these states. So the specific question here is, if you shift an organoid from basal to classical in culture and then transplant that organoid back into a mouse orthotopically, will the classical phenotype be maintained, or would it revert?
So it's a great question. We haven't done it yet. It's on the horizon to do. My guess is, because we see that some of the immune response actually drives it to the intermediate transitional phenotype, that's what is going to happen. And, the general theory that I have in my head, which is a theory, it's not validated, but it's the thing that we kick around when we're talking about this over coffee, is that, sort of you start in a more classical state, and that environmental factors, including, inflammation, drive you to a more intermediate transitional, which then can let you go into the basal. And so my guess is that you would transition from being more classical to more basal over time, and that would associate with worse prognosis. But I make no promises. That's just what we think, and we're going to try and do the PDX experiments to test that out.
Great, and you mentioned that I think it was the classical state was, I forget whether it was more or less sensitive to drug.
So the classical is more sensitive to gemcitabine, which is sort of the standard chemotherapy that's used in PDAC. And as you move it into the intermediate transitional by adding TGF beta, what we see is that actually it becomes about 10 to 100-fold more resistant to chemotherapy.
Right, and so is there any evidence that sort of transition accounts for resistance in human tumors?
Absolutely no clue, but that's what we're going to try and do. The only place where we have the longitudinal data in patients is sort of that N of 1 where we look pre and post immunotherapy, but it's definitely a place we're going. And it's a place where we're always super excited to partner with people at the Farber on doing longitudinal studies or doing trials where we can actually look and where we can create models and then try and think about drugs and combinations. And I think in a lot of ways, everything I showed you today opens up more questions than provides answers, but it tells us that we have to think very carefully about how we model the tumor as we think about drug selection, but it also, I hope shows you how much we can do even with an incredibly small biopsy that you wouldn't have thought about, using for single cell RNA sequencing and organoid generation and DNA phenotyping in the past.
Yeah, that's great. I mean, the SQL system seems really powerful. So I guess, when do you see it moving into actually clinical use, clinical pathology labs, and when?
So I would love to see it working. I'd love to see the platform move into clinical pathology labs. And, one of the reasons Chris and I started this company called Honeycomb is we wanted something that was simple, where it was a consumable that would work very robustly. It's less expensive to do SQL than it is to do some of the commercial variants, but it's also a little bit more of a pain in the butt because it's a homebrewed solution. And so what we're working on now, and better done in a company than in our own lab is, an injection molded plastic system that, sort of like snaps together and where everything becomes incredibly simple. And that uses the same freezing that I showed you. And so my hope is that it will not only enable, clinical studies where we can basically load cells and store them in these cellular hard drives to go back to later as, part of a clinical study, but will also enable you to engage with, clinics all around, all around as opposed to just here within Boston, because it would open up, where you could recruit patients and how you could do a lot of these things in ways that I think could potentially be transformative, or at least that's the vision.
Great. We have another couple questions from the audience as well. So Daniel Temko is asking how much of the within tumor subtype heterogeneity is due to clonal heterogeneity, such as subclonal KRAS? Have you explored whether different levels of amplification of KRAS or point mutations change the phenotype?
So it's a fantastic question. So, it's 23 and, maybe about 60% of the organoids took. So I don't want to, make a mountain out of a molehill. What I'll tell you is we see that the environment seems to dictate phenotype over genotype. We did show you that one example of, P63 amplification in addition to a couple other things where, really you had a strong genetic driver that really pushed you into a basal phenotype. We do see that even with, that we can have cells with a, with a KRAS amplification move from being basal back to classical. But what we really need to do now is we need to systematically explore how, different copy number alterations and how different mutations actually overlay on top of this. And that was what I was trying to get at really quickly with the ALL story, is that there seems to be this incredible coupling between genotype and phenotype because, the cell is sort of the machine that translates one into the other. And what we're trying to understand now is when and how are those mutations used, and how much does it lock you into a particular state, and how much does the environment decide which state is the most fit and therefore the one that you get locked into. And I think that as we think about how we might use environmental perturbations to shape cellular phenotype, it may give us a new strategy for making our drugs more effective, or for forcing cells into more liable, or into positions where they are more targetable, or at least that's the hope.
Great. And we're over time, but so I guess folks can leave if they want. I do want to ask two more questions, if that's okay.
Sure.
So one is from Wei Yang, which is basically, do the different TGF-beta isoforms 1, 2, and 3 have the same effects? Do you know?
We do not know yet. It is a place that we're going. What we're really trying to do now is we're trying to scale some of this stuff up so that we can figure out how to do screens, and the fact that we can generate the same sort of plasticity in the cell lines gives us another handle where we can start to test a large number of factors. And so in our future, I see a lot of screens, not only in cell lines, but also in organoids, and then hopefully in some resections. And that thing that I referred to very briefly at the end, where I said one place that we're really keen to go is into thinking about compressed screening. For us, this means looking at multiple different compounds on very limited samples. And so our hope is to actually start to look at the effects of all of these different things and understand if they're similar or different, to create more, let's say precision, not personalized, but precision medicines.
Yeah, that's great. And I'm going to ask the last question, which is, I think you probably have access to more single cell RNA-seq data than most. And you put that slide with PCA, to show sort of reduced dimensionality to that in that one experiment. Do you have a sense of how many dimensions there are in transcriptomic data? If you take all tissue types, all the cells that you explored, is the number of dimensions really 20,000, or do you think it's substantially less than that?
So it's a hard question. I mean, I think that, how many axes of variation are significant, it really depends upon the system that you're looking at. And as you add more and more cells, the number goes up. I'd like to say that it's fewer because there are conserved pathways and programs. But what I've learned from looking at, a lot of different systems is that you get strong dysregulation of some of the signaling networks because of mutations, or because of, because of translocations or other events. And what that means is that you get a rewiring. And so at a high level, you may see that, you find one significant axis of variation that associates with the signaling, and at another PC, you see a break that has to do with whether or not, all the genes are coupled, or if they've now become parts of two independently regulated programs. And so there's no simple answer, but I think that I'm hopeful from all the data that we've seen that you can reduce a lot of the data down to some major programs that encode basic cellular functions. And that, maybe we can't think about how to, go after specific targets, but we can start to think about targeting pathways, and that might be a more reducible space. And then it becomes figuring out which pathways do we need to target, and thinking about what are the best points at which to intervene in those targets to have as much of an on-target effect as we can while minimizing off targets. And so generically, let's say 30 to 50, if you need a number for the top of your head.
Yeah, great, excellent. Well, thank you so much. Yeah, very exciting stuff. I think any additional questions maybe could go to Alex's email. Hopefully see you again soon.
Thanks so much for having me and for joining.